Hello students, welcome to the program of Bachelor of Science 3rd year, subject Botany, semester 5, paper code BOC 107 and paper title Microbiology and Plant Pathology. Title of the Unit 2, Methods in Microbiology. The module name is Methods of Pure Culture and module number is 08. In this e-module, I'll be explaining the types of cultures, that is mixed culture and pure culture, and later I'll be explaining the different methods for isolating the pure culture like the streak plate method, spread plate method and the pour plate method. At the end of this e-module, the student defines the different terms, explains different methods, differentiates between different types of method and applies different methods for the growth of different microorganisms. How do we define culture? What is culture? It is nothing but the microbial growth. So whenever we grow the microorganism on the media, we call it as a culture. Culture is of two types, pure culture, mixed culture and pure culture. Mixed culture is a culture in which there is a growth of more than one species of microorganism on media. Whereas in pure culture, there is growth of only one species of microorganism on media. Now, when we want to isolate the pure culture, that is only one species of microorganism on a media, we follow three different methods. Number 1 is the streak plate method, number 2 spread plate method and number 3 is the pour plate method. Now coming to the streak plate method. In this method, we always use the inoculation loop or the inoculation needle. Inoculation loop is made up of a handle and a nichrome wire which is bent at the tip to form a loop. This loop is sterilized by the flame sterilization method and then it is dipped in a broth containing the microbial culture. The inoculation loop is removed from the broth and it will have the film of culture. Now, the inoculation loop is spread on the culture media in a particular manner which we call as a streak. Depending on the type of streak or depending on how we move the inoculation loop on the media, we subclassify the streak plate method into zigzag method, hexagonal method, quadrant method and the T-shaped method. In the zigzag streak method, we move the inoculation loop on a media in a zigzag manner. After streaking, we place this plate into the incubator at a required temperature for 24 hours. And after 24 hours, when the plate is observed, you will find that the microbial growth will be seen only on the strict area, that is, the microbial colonies will be present on the zigzag area. In the hexagonal method, the streaking or the inoculation loop with the culture is moved in 6 areas and it is later incubated at the required temperature for 24 hours and after 24 hours the microbial growth will be seen in the 6 areas in a hexagonal manner where streaking was done. In the quadrant method, the streaking is done in the 4 areas And again, when we incubate the plates after streaking at the required temperature for 24 hours, after 24 hours, the microbial growth will be seen only on the streaked areas, that is, in the 4 areas, means in the quadrant. In the T-shaped method, as you can see in the picture, the streaking is done in a T-shaped manner and the microbial growth will be restricted only to this streaked area. Now we come to the pour plate method. In the pour plate method, we take 1 ml of serially diluted culture or the sample in a sterile Petri plate. Later, we pour the molten agar. We mix the molten agar and the culture thoroughly by swirling the Petri plate. Or we can take 15 ml of media. To this, we add 1 ml of serially diluted sample, mix it thoroughly by swirling the conical flask, and then we pour the required quantity into the Petri plate. We allow the plate to solidify and then we incubate it at a required temperature and for 24 hours. After 24 hours, when you observe the plate, you will find that the microbial growth or the colonies will be seen within the media as well as on the surface of the media. The microbial colonies are smaller in size when they are within the media, whereas the microbial colonies are larger in size when they are on the surface of the media. Now coming to the spread plate method. In this method, we take 0.1 ml of serially diluted sample and we place it on a media, that is the sterile media. With the help of L-shaped glass rod called as spreader, we spread this culture or the sample all over the surface of the media. We incubate it at a required temperature for 24 hours. And then after 24 hours, we see the microbial growth all over the surface of the media. Now, this particular method is generally used for the enumeration or the quantification of the microbial colonies, whereas the strict plate method is used to isolate the pure strain of microorganisms. Now, whenever we follow these 3 methods, we always get the pure culture, that is, we get the growth of only one type of microorganism or only one species of microorganism on the media. And thus, whenever we want to isolate the pure culture, we follow either of these 3 methods. These are the few references which you can refer to for preparing this module in detail. Thank you, students.